Okay, we're going to go over a couple of questions from this most recent quiz as well as some quizzes previously. And the first question we're going to go over is about the template used in PCR. So the template used in PCR, is it the nucleic acid that will be amplified? Is it the sequence that Taq polymerase recognizes in order to bind? Is it synthesized artificially and added to the PCR reaction, or all of the above? Now, the reason we're going over this is a lot of people answered this question with this answer, which is incorrect. And the reason it's incorrect is the template DNA is not synthesized artificially. What it is you have your double-stranded DNA, you have your primers, your forward primer and your reverse primer. The primers are synthesized artificially and added to the PCR reaction, not the template. Primers are made using a DNA synthesis machine. You usually order them, you pay for the primers, the cost is based on the length, and you'll use the primers that are synthetically made. But your template DNA is DNA that you extract from a cell, so it's not synthesized artificially. So that's why this answer was incorrect. Now, the template, is it the sequence that Taq polymerase recognizes to in order to bind? And that is also not correct, and it's because Taq polymerase doesn't actually recognize or bind to the template. So Taq polymerase is different than restriction enzymes, where restriction enzymes recognize a specific sequence. Taq polymerase does not. Taq polymerase will make DNA by adding nucleotides, complementary nucleotides, to copy a strand of DNA. So Taq polymerase just makes DNA. So the correct answer for this was this answer. It's the nucleic acid that will be amplified. That's what the definition of the template. The template is what you are amplifying. Now, what is complementary DNA? Is it DNA made from an RNA template? Is it the strand of DNA opposite the strand used to make messenger RNA? Is it the sequence of a cloned DNA? Or is it a sequence of DNA as it is found in the genome? Well, complementary DNA is not DNA found in the genome. In the genome, in your cells, in a human cell, in the genome, you have DNA in your genome. Is the sequence of is complementary DNA the sequence of cloned DNA? And we talked a lot about cloning today in week 10, and the complementary DNA has nothing to do really with cloning, so that is not correct. Is it the strand of DNA opposite the strand used to make messenger RNA? No, that's not what it is. So what complementary DNA is when you have messenger RNA and you use an RNA polymerase to make a DNA copy of the RNA. That is what complementary DNA is. So this was the correct answer here. So it's not really opposite the strand. Messenger RNA doesn't have anything opposite from it. It's just there waiting to be translated into a protein, and you can isolate the RNA from a cell and then use RNA polymerase to make complementary DNA off of it. Okay, there's some math problems that there were some issues with, And one of these math problems is here. I changed some of the numbers, and it reads as a DNA sample that you recently extracted in a total volume of 500 microliters has a concentration of 80 micrograms per mil. What is the total yield in micrograms of this DNA? So what you want to do here is first convert your microliters into mils, and that is because you have your concentration as 80 micrograms per mil, and you are going to then multiply by the volume, and here you have mils, so you want your volume to be in mils here. So 500 microliters equals 0.5 mils. So that's what you need to put here, 0.5. If you take your calculator, you should do 80 times 0.5, and that will give you 40 micrograms is the answer to this question. Another question is, calculate the total yield of RNA in micrograms again from a 0.2 mil sample diluted 1 to 100 with an absorbance reading at 260 of 0.250. So you remember your total yield is your concentration times your volume. So you have your concentration is the reading at 260, which is 0.250, times your factor for RNA. If you remember, that factor for RNA is 40 micrograms per mil, times your dilution, and your dilution here is 100. So you now, that is 0.250 times 40 times 100. This is 1,000. And then you multiply it by 0.2 mils, your concentration times your volume, to get your yield. So this is 200 micrograms. So that's the final answer for that one. Now another question: after completion of an agarose gel electrophoresis of DNA, along with the proper molecular weight standard, the gel is blank. So in other words, you have an agarose gel. You ran, let's say in lane 1, you ran your marker. In lane 2, you ran your controls, positive control, negative control, sample. There are no bands at all. None. You stain it and there's none. So, well, I said maybe a possible answer. What's the most likely reason? So here are several reasons. If you get a completely blank gel, one possibility is that you did not stain. You forgot to put it in stain. If you don't stain it, if you don't stick it in ethidium bromide, you're not going to get any DNA to show up. So that's one reason you might not get any bands at all. Another reason might be you forgot to load anything. You forgot to load any sample into each well. So if you forgot to load, your gel is going to be completely blank. So those are really the two main reasons why you would get absolutely no bands on a gel. One of the possibilities that was an answer on a similar question on a quiz was that the DNA was degraded. And That's not the right answer. And the reason that's not the right answer is you have your molecular weight standard. Your molecular weight standard has nothing to do with any type of reaction. I mean, your molecular weight standard should run. You should see it. If you stained your gel, you should get bands for your molecular weight standard. It would be very unlikely that all of your DNA, all of your samples, all of your controls, and your molecular weight marker all degraded. Also, if you have DNA in this lane and it's degraded, usually you're going to get a smear of degraded DNA. You'll usually see something. It just doesn't show up as nice crisp bands. So the reason that you would get absolutely no bands at all, including no molecular weight marker, is you forgot the ethidium bromide or you forgot to load your samples into your gel. So these are just things you really have to think through when you're reading these questions. A lot of times what I like to do is read the question and think through it first. Have a scratch piece of paper next to you, write some possibilities out. Don't even look at the answers, because sometimes looking at the answers can be confusing. So if I can figure out the answer just from looking at the question on my own on a scratch piece of paper, and then compare my answer to what the possibilities are, and I see a match, woohoo, then I know it's most likely that I have the correct answer. So that's just a little tip when you're taking some of these multiple choice exams.